Ever wonder if you could use your DNA to stop picking the wrong one for you? So what exactly is DNA, you might ask? DNA is a nucleic acid which contains all of our genetic instructions. Our cells decode it and use it to develop and carry out functions as a living organism. The four main functions is to replicate, encode information, mutation and recombination of genes, and gene expression. Over many years, numerous scientists worked to reveal the structure of DNA. James Watson, Francis Crick, and Rosalind Franklin were awarded for its discovery in 1962. James and Francis came to the groundbreaking conclusion of the structure, but they could not have done it without Rosalind's X-ray images of the molecular structure. DNA consists of a double helix, which makes it look like a spiral ladder. The phosphated sugar molecules form the ladder's backbone, and the nitrogenous bases form the rungs. Each unit of DNA is composed of deoxyribose sugars, phosphates, and nitrogenous bases. This is the formation of one nucleotide. Each nucleotide is about 2 meters long and in every single cell. Humans have about 3 billion nucleotide pairs. Erwin Chargaff is to thank for discovering the 4 types of nitrogenous bases found in DNA, which are called guanine, adenine, thymine, and cytosine. They bond together through hydrogen bonds. Adenine always pairs with thymine with 2 hydrogen bonds, while guanine always pairs with cytosine with 3 hydrogen bonds. Adenine and guanine are called purines because they have double rings, while thymine and cytosine have a single ring, so they are called pyrimidines. Deoxyribonucleic acid versus messenger ribonucleic acid. DNA is formed by a double helix that consists of adenine, guanine, cytosine, and thymine, while mRNA is a single-stranded and it consists of adenine, guanine, uracil, and cytosine. Thymine in DNA becomes uracil in mRNA. Since our DNA cannot leave the nucleus, it makes a copy called mRNA. This process is known as transcription. The mRNA is allowed to leave the nucleus where it meets with ribosomes, our building proteins within our cells. The ribosomes read the information and decode the codons to the amino acids, which forms a polypeptide chain, giving us all of our features, characteristics, and basically our entire identity. One of the most relevant debates in biology is that of nature versus nurture versus epigenetics, which is our DNA versus how we are raised versus DNA altering. When studying this debate, scientists often use identical twins to see how nature and nurture plays a role in their lives. They found that even though they have identical DNA, if they are raised in very different environments, they will become different from one another. However, since their DNA is the same, they will still have similar health risks and personality traits. However, how they are triggered and when they come up to be depends on their environments and their diets that will differ. Epigenetics is altering DNA in the womb, giving your child desirable features. It works by inserting chemical tags onto the chromosomes to turn some genes on or off. An example is DNA methylation, which is the addition of methyl groups to part of the DNA molecule, which prevents certain genes from being expressed. The Human Genome Project began in October 1990 when the plan to map the entire genetic makeup of a human being began. With the aid of scientists globally, they completed the project in May 2000. The human genome consists of 30,000 genes within 23 pairs of chromosomes, and it is found within every single one of your cells. The main reason for this project was to identify genetic disorders, allowing scientists to then look for cures in medications and in gene therapies. Although this project does seem very beneficial, it does come with a lot of ethical issues. It will advance science and help in the fight against genetic disorders, but it will also bring rise to a lot of societal problems and legal issues. Who will be able to get this information? Your bosses, government, the police? This can cause many issues for you, especially when it comes to finding work or even a partner. Although gene therapy is still developing, there are currently 3 strategies. The first being inserting a normal gene through a virus, the second being attempting to chemically recode the genetic message, and lastly, gene surgery, which must be performed on a zygote. DNA sequencing is determining the exact order of the 4 chemical bases that make up the DNA molecules. It is a great tool to use to find out if you have any problems within your cells so that you can take control early on. There are many companies that can do this for you, such as 23andMe. This brings us back to the question of the video. Have you ever wondered if you could use your DNA to stop picking the wrong one for you? Well, you can. By sequencing your DNA, you can get a better understanding of your immunogenetics, which has a strong effect on relationships and compatibility. The idea of opposites attract is true when it comes to our immune systems. Dating websites have been created, such as Pheromore, which uses your DNA to find the perfect match for you, thanks to Brittany Barreto, Asma Mirza, and Bin Wong, who swear to only look at certain genes that have any connection to who you can be attracted to, and promise to keep the rest of your genetic information safe. They collect your genetic data through a cheek swab that you send them. They single out 11 genes and swear to only sequence and look at those 11 genes. These genes are linked to your pheromones, which are chemical signals in our bodies that allow us to be sexually attracted to something or someone. It was discovered that humans find people with opposite immune systems to be more attractive, and they are more attracted to their scent. This is studied through MHC genes, which influence our sexual desires and are linked to our height and scent. If you're sick and tired of wasting time on the wrong one for you, the next time you go on a date, bring them a cheek swab and you can find out if you have opposite immune systems and see how attracted you really are to the person. I do believe using your DNA to find the right one for you is a great way to do it. Especially using websites such as Pheromore, you can find people all around the world that are the perfect match that you wouldn't find in your day-to-day life. There are also many other pros and cons. If you intend on procreating, you'll find out what kind of genes your children will be inheriting, and you'll also be able to tell your mother if you found the one and you're not just wasting your time. However, it is a lot of information to know on the first day, and you probably won't be making it to a second one.